Okay, so now we're going to start talking about how RNA is processed after we've made, as we are making our messenger RNA transcripts. And so we're going to describe, for example, the 5' cap. We're going to describe different types of introns and how they are spliced out. And then we're going to discuss how the RNA message is processed and split into potentially even multiple different proteins. So the processing are going to be typically catalyzed by our RNA, so those ribozymes. So introns are going to be spliced out by ribozymes, by enzymes. And the RNA molecule is going to be a primary transcript. And in eukaryotes, this primary transcript will contain one gene. And as we talked about with bacteria, their primary transcript contain multiple genes. So in essence, our primary transcript is going to be, template is by the DNA, and then you're going to get at the 5' end a cap, and this cap is added as the gene is being, or the message is being made. And then eventually we're going to get a 3' poly-A tail added to it as well. But what's important is that post-transcriptional modification is going to include all of these processes. We're going to have to splice out, for example, our introns, and we're going to have to add our poly-A tail, so polyadenylation. And then eventually we can have different cleavage sites as well, which we will discuss. Now, our 5' cap is actually very interesting. Our 5' cap is going to be a unique linkage between our 5' end of our RNA. So this is the 5' end. We have that base. And then we're going to want to add guanosine. And this is a special guanosine. It's been methylated at its 7 position, shown up here. And this guanosine is going to attach to the 5' end of our messenger RNA at their 5' carbon in the ribose. And so this is a 5' terminal of the messenger RNA linked to a 5' residue of a 7-methyl guanosine. And so where did this guanosine get its methyl group? Well, it's got its methyl group from SAM. So basically, SAM, which is a single carbon donor, which requires vitamin B12 plus folic acid, will provide the methyl groups to add to our guanosine for our 5' cap. And so again, this is another point where SAM is going to be very important. And SAM is S-adenosyl methionine, so it also needs methionine, is going to be very important in providing the caps to protect our 5' end of our messages. Now, the whole purpose of this 5' cap, again, is so that the message doesn't get degraded by enzymes in the actual cell. Now let's just talk a little bit about our ribozymes. So our introns are going to be spliced by RNA catalysts, also known as ribozymes. And so this is just a funny picture I found that kind of displays this. So you have some sort of target, and then your ribozyme will sort of bind to that target. And as we saw with DNA, it's the same thing with RNA. It's not just very precise binding. The binding is going to be upstream and downstream of the actual target. And so the binding of the ribozyme is going to be upstream and downstream, but it's going to identify some sequence where it can cleave. And then you're going to get 2 products from that cleavage. Oops. So here's 1 and here's 2. And so those 2 products from the cleavage are going to be what the purpose of that ribozyme were. In the case of introns, you're going to splice out introns, and then you're going to want the exons to be brought together. Now, ribozymes are also going to be important for processing of our transfer RNAs. They are also going to be important for protein synthesis, and we'll talk about the peptidyltransferase ribozymes when we talk about protein synthesis. And also they can chop up messenger RNAs, so they're going to be regulatory as well. So there are going to be 4 classes of introns. We have group 1 and 2 introns that are going to be self-splicing. So this means that there are no enzymes involved. They are their own splicing units. But what's really important is that this is not most of our enzymes. Most enzymes are not self-splicing in eukaryotes. Now, group 1 is going to be important for ribosomal RNAs, transfer RNAs, messenger RNAs. Group 2 are usually going to be mitochondria and chloroplasts. So these are going to be for our cells in the mitochondria. And again, there is no ATP required. These guys are self-splicing. So these are themselves ribozymes. And so they're going to have a 3' OH, which is a guanosine, and we'll talk about that. So guanosine is going to be a nucleophile, and that's going to attack the phosphodiester bond. And so because guanosine is the actual nucleophile, shown here, there is no energy lost or gained. So for a group 1 transcript, we're going to basically have the guanosine, this is the attacker, and the PG can either be GTP or GDP or GMP. It does not matter. And so that phosphate guanosine is then going to be, that OH group is going to be the nucleophile, and it's going to be specific for the 5' end. And so that 3' hydroxide group of guanosine is going to form a phosphodiester bond with the 5' phosphate at the 5' end of the intron. And then you're going to have your 3' OH is going to be displaced, and this is going to act as a nucleophile for the 3' end. And then finally, you're going to get the intron with the 5' guanosine at one end and that OH group at the 3' end released. And this brings together those 2 exons, and they're able to come together and form a bond. The intron will be degraded. And again, this is a very simple splicing out of introns that is done with no energy required. Now, group 2, the intron contains a nucleophile itself on an adenine. So here's the adenine with the OH group. And so the primary transcript in this intron itself has the nucleophile. So adenosine is going to then take on a 3' phosphodiester bond, and it's going to be referred to as a Lariat structure. And then the Lariat structure is going to have 3 phosphodiester bonds, and the 3' end of the the OH group, at the 5' end is going to be a nucleophile that's going to attack the 3' end. And this will release the Lariat structure. And this is a group 2 intron splicing. And again, it has its own nucleophile within it. It's its own, in essence, ribozyme. It does not need anything external to help it. And this is going to be done in eukaryote cells in the mitochondria. Now the next group is a group 3 splicezyme, and these are the more common in eukaryotes. This spliceosome is going to be required to cleave out these introns. And the spliceosomes are going to have many different proteins associated with them. We're going to have our small nuclear RNAs, and there are 5 small nuclear RNAs that are going to be important: U1, 2, 4, 5, and 6, shown here. Now the splicing mechanism is basically the U1, which is shown here. The U1 small nuclear RNA has a sequence that is complementary to the sequence at the 5' end of the intron. So U1 sequence. okay, has a sequence. U2 is going to have a sequence that pairs with the region with the adenosine, and this will be the nucleophile. So this adenosine, when it's bound to U2, is similar to what we saw for our type 2 intron splicing mechanisms. So U2 binds to adenosine. And this is going to, of course, cause eventually a bulge. Now U1 and 2 will bind, and then this will recruit U4 through U6 complex. And then U5 will bind, and it'll become an active complex. Oh, here we go. And so first, of course, we have U1 that binds that 5' end. We have U2 that binds the adenosine. You get recruitment of U4, U6. This requires ATP, so energy is required for this. U4 and U6 are going to create sort of this bulge for this intron, and then U5 will be recruited. Now this is considered the inactive splicezyme. And then you're going to have some rearrangements internally that will cause the spliceosome to be active. So the inactive spliceosome is going to kick out U4 and U6, so these get released. And then now you have your U2, U5, and your U6. Now, your U6 is going to pair with the 5' end of the intron, and it's also going to pair with U2, so that's what expels the U1. And then splicing will occur similar to a group 2, the formation of a Lariat structure. So U6 binds the 5' end, and U2. And then you have a Lariat that forms. And the Lariat forms because you are using that adenosine. So you are using this adenosine to form that Lariat structure. And then you'll get release. That OH group is going to become the nucleophile. So you'll get your exons brought together, and you'll get release of your intron. And then the intron will be degraded. So U1 and U2 were those first that bound, and they are going to target a 5' GU and AG3' introns. There are going to be other introns that are going to be targeted by other small nuclear RNAs, but they're a little more rare. But there are other proteins. There are, for example, U11 and U12. They will target a different sequence of 5', 3' introns. But the most common is the one that is targeted by U1 and U2. Okay, so now the type 3 introns are going to be spliced as they're being transcribed by polymerase II. So the components of the spliceozyme are going to be attached to the polymerase. And it's all going to occur again at that C-terminal domain. And so the splicing units will be tethered to polymerase II. And then as the intron comes out, those splicing units will bind to them. They will capture the second one coming out, whatever that is, depends on the adenosine. And then you will get the removal of your intron. That Lariat structure will get removed, and the exons will be fused together. So again, this is occurring as part of the function of your RNA polymerase. Now, group 4 introns are going to be associated with transfer RNAs, and these are going to be Similar to group 1 and 2, except for it requires ATP, and you do have an endonuclease necessary. So you're going to cleave basically the phosphodiester bond at both ends of the intron, and then you're going to have ligase that's going to have to bring the exons together. Now, after you get your message made, your introns removed, the end has to be protected, and it's protected through a process called polyadenylation. And this is going to add, at that 3' end, adenosine residues. And again, similar to the 5' end with the cap, this 3' end is there to basically protect the messenger RNA and eukaryotes against destruction. Now, this has an alternative process in bacteria because polyadenylation will actually target the message for destruction. Now, again, this is all going to happen at the C-terminal domain. So we have the C-terminal domain again. And you're going to get what's called polyadenylation factor that's going to attach at that region. And this signals sort of the end of the transcript. And so what's going to basically happen is that your polymerase is going to be released through an endonuclease reaction. Your message is then going to be attached with that polyadenylation factor, this little purple blob. And this is going to recruit your polyadenylate polymerase. And then your polyadenylate polymerase is going to be responsible for adding the tails. Now, the transcript to polymerase 2, the transcript that is going to be upstream that recruits the polyadenylation factors is going to be a 5' AAUAA sequence. And this is bound by the enzyme complex. And this is going to contain this complex will contain both the endonuclease that is going to release the polymerase, and it's also going to contain the polyadenylate polymerase. Now, all enzymes and that 5' AAU-AAA sequence are going to be tethered to that C-terminal domain. So again, similar to the 5' cap, the polyadenylation is going to take place associated with the polymerase 2. Now, the polyadenylate polymerase is going to synthesize the poly-A tail. It's going to add 80 to 250 nucleotides, beginning at the cleavage site. So you get 80 to 250 A's attached. Okay, so this is just an overview. This is albumin gene, similar to something like serum albumin. And so we have in here, we have our leader sequence, which is this L here. And this will tell the protein where it needs to go after it's been translated. And so the leader sequence for ovalbumin will tell it it needs to be secreted from the cell. Now the ovalbumin is going to have several introns that are going to get spliced out. And it also has a little bit of extra RNA that needs to be sliced out. And then the mature messenger RNA is going to contain that 5' cap, and it's going to contain the poly-A tail and all of its exons in between. Now, that's a pretty straightforward processing of RNA. So we have some RNA that are going to have one message, one protein. Some RNAs will have one message, multiple proteins. And oftentimes the difference is in the location. So where is the messenger RNA being expressed? So sometimes you have a transcript that will be expressed in the gut and another that will be expressed in the brain, and they have two completely different products based on their location. So when multiple proteins are possible, the one favored depends on the specific cell's processing factors. So an example is you can have multiple sites for polyadenylation. So here's our polyadenylation sites. we have one site that's A1 and one site that is A2. If you're in a cell that chooses A1, your messenger RNA is going to be shorter than the cell that chooses A2. You're going to have a longer transcript. And so this is basically just going to lead to two different products in two different cells based on the factors within that actual cell. So again, that gets back to that whole concept of epigenetics. Now you can also have different 3' splicing sites. The 5' end doesn't change, but the 3' end, because remember this will have eventually an OH, and so this can attack this 3' end, or it can attack this 3' end. And so again, you're going to have differences in the size of the transcript. One is going to be much longer, and then one's going to cut off more and be much shorter. And again, this is going to happen in different cells depending on their different components that are going to be involved in splicing out these introns. Now, an example that is used is calcitonin in rats. So here again, we have our primary transcript. It's going to have our 5' cap, our polyadenylation tail over here. It's going to have our introns. It's going to have two important genes that are necessary in two different locations. We have calcitonin, and then we have the calcitonin gene-related peptide. And so in the thyroid gland, you're going to want to get calcitonin. So that means you're going to start with this first polyadenylation site. So you're going to get a smaller transcript or different transcript. In the case of your brain, you want a different protein made. You want the calcitonin gene-relating peptide made. And so this is going to require the alternative polyadenylation site. And so the mature messenger RNA will be different and will contain that protein that is necessary for the brain.